ribosomes, new storage granules, all of that good stuff that you have, you need to make enough of it so there's now enough for 2 cells. So for instance, right, you need ATP for the reproduction of offspring, which means also that you need to manufacture cellular components, right? But we have this thing called entropy in the world, right? Which means everything tends toward chaos, which means stuff breaks, right? And as you age, you'll experience that and you'll know what I mean, right? Stuff wears out. So we have to repair things, right? And it takes energy to reproduce, it takes energy to make new parts. So even if you're not reproducing, you still need ATP. So you need to make new things, you need to repair things, right? But this process, what we're going to be studying in this chapter, binary fission, which is shown here in a transmission electron micrograph. How can I tell? Number one, the cells are really, really big. So I have to get really, really close to them, which is what a transmission electron micrograph does. Another thing that would tell me that is that I'm seeing through them, right? Electrons have been transmitted through them. I see a flat image, right? So to make this happen, we need new ATP. And to make new ATP, or sorry, I need to make new DNA. To make new DNA, it takes about 10 ATP per base pair. That's a lot of glucose just to make, right? So think base pair. Okay, well, how big a chromosome could a bacterium possibly have? E. coli's is 3 million. So that's a lot of ATP just to make another chromosome, right? Well, what about the genes and the proteins that the DNA is used for? Transcription, about 5 nucleotides per RNA. That's a lot, right? How about translation? Translation, at least 3 ATP per amino acid that gets added to the amino acid chain, right? I'm not going to ask you, but just, right, the costs, growing costs, right? What about making your cell wall? Cell wall, just for one of these bricks, it'll cost you 4 ATP per brick. So this process of binary fission, do not memorize those. Don't waste the brain space. The reason we're looking at them, I see you all madly saying, don't change the thing, I haven't written it down yet, right? It costs ATP, right? So this process of binary fission, where we start with one cell, we duplicate everything inside that cell, and we split the cell in two, it costs. So 5 and 6 are intimately related to one another because chapter 5 says, where do you get your ATP? And chapter 6 says, this is what the majority of that ATP is spent on, because unlike you, bacteria, as soon as they are able, reproduce, and they reproduce as quickly as they can forever until they die. So for E. coli, that means about every 20 minutes, as soon as he's born, 20 minutes later, he undergoes this process, which is really phenomenal if you think about the fact that he can be a cell, grow all this, like take in enough food, grow everything, and split every 20 minutes. And you know he's not the fastest. There's an organism that causes some delightful diarrhea. His name is Clostridium perfringens. You don't want to get him. And he can do this every 7 minutes. It's pretty crazy, reproducing every 7 minutes, right? Can you imagine if you got pregnant and 7 minutes later you had a baby? That'd be a little bit crazy. Maybe that'd be nice, right? I wasn't a really big fan of being pregnant. I love my kids, but I don't like being pregnant, right? But we have a much longer period of time, right? So 20 minutes for E. coli, that's pretty standard for the things that we work with, which is why we can put them in the incubator. And even when we have summer classes and we come back tomorrow, they're grown by tomorrow morning, right? Because they grow quickly. This process is very involved, but all we're going to say is, you make another copy of your DNA, you get a little bigger, you put your dividing wall between you, and boom, now there are 2 of you, you're genetically identical to one another. It is not mitosis, because mitosis technically is nuclear division. And what do we not have in prokaryotes? A nucleus, right? So it's not, but it's exactly the same. If you think about that whole like doubling everything and splitting, right? 1 cell, 2 cells. Now, I don't know if you remember, but when we were in chapter 4, I said your book irritates me because it shows the DNA sort of floating around in the cell, and I said, and there's going to be a picture later on where it actually, they show the DNA accurately attached to the inside of the plasma membrane. Because think about those tumbling cells you saw, right, with your wet mouse. Tumbling, right? Imagine tumbling, your DNA is tumbling and it's getting banged around and your DNA breaks, and if you don't have DNA, you don't survive. So one of the ways that a cell who cannot enclose their DNA in a nuclear envelope protects its DNA is by attaching it to the plasma membrane. And that also explains for us why those bacilli that we learned about earlier on cannot make staphylo arrangements. Right, remember we talked about bacilli, and I said bacilli can only divide like this, which means they can only make diplo, right, and then 4, or, right, they might stay attached and, or make diplo, and they might stay attached and make strepto. But they never make tetrads and they never make staphylo, right? That's not possible. Now, you might have been thinking, right, if you're a stickler for this whole symmetry thing, you might have been thinking, well, okay, I mean, they're bilaterally symmetrical, this way and this way. But that's only from the outside. From the inside, look at these two pieces of DNA. The only way this cell is symmetrical is by dividing it on that plane right there. Right? If I take and divide it on this plane, I will end up with 2 pieces of DNA in one and none in the other. And that is the underlying reason why you can only ever have single bacilli, diplobacilli, or streptobacilli. You can't have staphylo or tetrads, because if they happen to do that, you would end up with 2 non-functional cells, right? So you might think having extra DNA is great. Well, a person who has Down syndrome has one of the very smallest chromosomes, an extra copy. Is that great? That's not great. Having too much is not a good thing. So having 2 copies of DNA is not a good thing. Having no copies of DNA is not a good thing. What you need is 1 copy. And the only way to get 1 copy is to divide down that portion right there. Now with the cocci, they can attach it in different places. But the bacilli attach it right in the middle of that bacillus shape, right here. Whereas the cocci, they can sort of attach it wherever they want to on the cell. Yes?
What's the advantage of dividing but still being connected? Like, why not be individual?
I don't know. Yeah. I don't know. I would think that being separate would be a benefit because you have space around you. But we're going to learn in chapter 8 there are lots of behaviors that bacteria turn on only when they're crowded. So in that case, it would be nice to have your buddies around you. I mean, if you're thinking totally self-servingly, it's good to have them around you, because let's say you're spraying calcicide on the countertop and there are bacteria around you, especially if you're in a staphylo arrangement, and they'll all die and you're protected from them, or by them, from the disinfectant. But I don't know why. Why that would be. I mean, I don't know particularly what advantage. I could just speculate. So binary fission is the process where one cell, right, splits into 2. So we call it binary fission. So binary fission starts with the cell elongating and replicating the DNA. This plasma membrane and cell wall start to grow inward, right? So this could be an example of a diplobacilli that stopped there. They're 2 distinct cells. They're both bound by a plasma membrane. But in this instance, like E. coli, the cell is genetically programmed to divide completely and break apart into 2 single bacilli. So you can see here another transmission electron micrograph. You can actually see the cross wall, right, the cell wall forming between them right there. So it forms from the 2 sides, much like a plant cell does, right? When you look at mitosis in plant cells, if you look at them under the microscope, you can catch them in stages of mitosis and cytokinesis and see that cell wall forming. So binary fission means that every cell will divide and become 2, we call them daughter cells, right? So daughter cells is a word that we use that tells us those are genetically identical to the initial cell. And the fact that they can grow and divide into 2 means that all bacteria grow exponentially. because the word exponential means to double. So they double with every generation time. 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, 32 becomes 64, and so on and so on and so on. So they can have really, I mean, not as many offspring as some other things, right? So you might think, ooh, they can double at every generation time. Like that means they can grow really quickly, right? Well, think about a rabbit, right? A rabbit can have like 8 babies at once, which is way better, right? That's 4 times as many offspring as a bacteria, right? Because bacteria can have 1, rabbits can have 8. The thing about bacteria having what we call biotic potential, the ability to grow quickly, does not rely on the fact that they grow exponentially, and that's a common misconception. Because they double every time they grow quickly. It's not that, it's the short time it takes to double. The generation time is what we call that. The time between that one cell becoming established as its own cell and then dividing to make 2 cells, right? So if we take E. coli replicates or undergoes binary fission every 20 minutes, which means it can do it 3 times an hour. It's that 3 times an hour that'll get you, right? Because if you double every 20 minutes, you will undergo 20 generation times in less than 7 hours. You know, 20 generations, it's not a big deal, right? Well, go to 20. 1 cell has become over 1 million cells. That's biotic potential, not because they divide, right? It's not the fact that they divide that gives them tremendous biotic potential over, say, you or another multicellular organism. It's not that, it's their short generation time. Because they do that generation so quickly, they have tremendous biotic potential, right? As compared, let's say, to Mycobacterium leprae, which causes, what do you think? Leprosy, right? It's incredibly slow growing. Its generation time is like 3 weeks or something like that. Don't quote me, I don't remember, right? Not everything up here is ship-shape. I mean, it is ship-shape, but I don't remember things. Like, don't say, oh, 3 weeks, 21 days exactly, right? But it's really long. So 20 generations for him is a much longer period of time than for E. coli, which explains why if you eat a bad salad with E. coli on it, you will get sick within like 8 hours. If you contract leprosy, it can be 10 years before you show symptoms because it grows incredibly slowly, right? So leprosy does not have the biotic potential that E. coli has. E. coli does not have the biotic potential that C. perfringens has because of the generation time. All of them divide by binary fission. It's the generation time that makes them have biotic potential, which makes them grow quickly. So one of the things that we study in microbiology is how bacteria grow, population-wise, right? We don't ever talk about bacteria growing in size. Like there's not like baby bacteria and adolescent bacteria and adult bacteria and old person bacteria, right? There's just bacterium dividing into 2 bacteria. So when we say growth, we don't mean growth of a single cell, we mean population growth. Because we never really work with one cell, right? When you inoculate cells into media, you have no idea what's going on until you grow them and you bring them back out, and then you see them, right? So we always talk about the growth of a population. And so when we study bacterial growth, we oftentimes study how populations grow. And this will maybe look familiar to you because bacterial populations grow just like any other natural population grows. All natural populations go through 4 phases of growth. And they are lag phase, not log phase, exponential phase. So this red, that's mine. So if you have this picture in your textbook, draw a line through log and put exponential. So they start in lag, they move to exponential, and then to stationary, and then to death phase. Every natural population on the planet. So a natural population of bacteria could be the loop full of bacteria that you inoculate into a test tube and then put in the incubator. So when you put those bacteria into the incubator, they are in what's called lag phase. lag phase from the outside seems very boring. Because if we look at that population over time, it doesn't change number, right? Over time, because time is on the x-axis, the number of bacteria over time stays the same. And you're like, well, that's boring, nothing's happening. You would be very, very wrong to think that nothing is happening there. What is happening there is they splash in and they go, oh, there's so much good stuff to eat. They start eating it, they start breaking it apart in glycolysis, tearing it down in the Krebs cycle, moving the electrons into the electron transport chain, making ATP. And they need that ATP to make DNA, to make the cell wall, to make, to transcribe mRNA, to translate mRNA into proteins, to do all of that work. And then all of a sudden, they've done all that work and they divide. And one cell becomes 2, and 2 becomes 4, and 4 becomes 8, and 8 becomes 16, and 16 becomes 32, and 20, blah, 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 blah. And they double over time. And that's why that phase is called exponential, not log, because exponential says doubling over time. So they double over time. So really, as much is happening here as is happening here, we just can't see it. If you could get inside the cell, you'd be like, holy cow, right? Like I'd be like, zoom, 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 right? Everything would be all around you, right? From the outside, though, you can see when they divide because we see the number of cells increase. And we see that happening over time. Cells are dividing, cells are dividing, cells are dividing, cells are dividing. They're eating, they're doing all the stuff of chapter 5, and now they're dividing, and now the food starts to run out because they're just in a test tube. And the food starts to run out, and it starts to get crowded, and all the waste that's being produced, right, metabolically, because they're growing, right? I mean, bacteria don't pee and poo, but they produce waste, just like you do. And that waste, right, it doesn't get, there's nobody that comes and takes the trash away, right? There's no flushing of a toilet to take the waste away. They're sitting in it. Imagine how long you would survive if you sat in your waste. Not very long, right? So as they grow, they create waste, they use up all of the material, and they start to slow down. And now, there's no change in cell number over time. So again, right, we look at this graph and we go, well, this looks the same as this, but you'd be very wrong, right? Whereas in the lag phase, lots of stuff is happening, all of chapter 5 is happening. That's very different than up here. Stationary phase, the name describes it, right? Stationary phase, it's kind of going into a stasis. Everything's kind of slowing down because waste products are starting to accumulate, food is becoming scarce, and they're metabolically much, much slower. There still is binary fission going on, it's just slower, and for every cell that's dividing, somebody's dying. So the population size is staying the same. And then, because you're in that test tube, more waste starts to accumulate, food gets even more scarce, and now more and more bacteria are dying, and that outnumbers the cells that are undergoing binary fission. So if we look at this, right, if I pull this up, here, right, the graph tells us that the rate of binary fission is equal to the death rate, right, because there's no change in the number of cells over time. But underlying that is a ton of activity, right? The activity level is through the roof, tons of activity going on. Then finally, all of their energy has been, right, they've done all their stuff, and at the end of that process, cell division happens, and now this says that binary fission is really high, right? It's much greater than the death rate. And there's still lots of activity happening. Every 20 minutes, cells are dividing. They're dividing, they're growing optimally, right? And then they hit the stationary phase, and from the outside we can say, well, if this line is straight, it has a curve of 0, that means binary fission is again equal to the death rate, but this and this are very different, right? Whereas we had a lot of activity here, we have very low activity level. And then over here, we get almost the opposite of the exponential, right? So now our curve has a negative slope. And now, binary fission is less than the death rate. So we, as we measure, see fewer and fewer cells over time. So we want to keep in mind that though this and this look the same, right, very different things are happening in the population. So when we study bacteria, one of the most important things that we can do when you're setting out to study bacteria is figure out how to grow them well. Like when I was in graduate school, my graduate work was on a particular bacterium from Mono Lake. I think I mentioned that, right? So one of the things that I wanted to do was figure out how to grow him well, because my whole thesis relies on him growing and me collecting data. So I needed to figure out how to grow him best. And I did that by manipulating the environment and watching him grow over time. And what I'm looking for is for this right here to have the steepest curve possible, because that means he's growing faster, right? This means not growing at all. This means growing relatively well. This means growing really well, and this is what you want, right? And in order to see that, you have to grow bacteria and look at them grow over time. You have to be able to count them. In lab on next Thursday, so not Thursday, but a week from Thursday, you are going to be doing a lab where you watch a culture, a population of bacteria grow over time. And we're going to try to determine the generation time of those bacteria by watching them grow. And by determining the generation time, we can determine the biotic potential, right? Because if the generation time is small, big biotic potential. Generation time long, smaller biotic potential, right? So we can do that in a couple of different ways. We've chosen two ways to watch a population grow over time. So we will look over time, and we will measure something from which we can infer bacterial growth, right? If something increases, then the bacteria are increasing. So in lecture, we are going to talk about 4 methods. In lab, we are going to perform 2 of them. So for lecture, you'll need to understand 4 methods. For lab, you'll actually perform and need to understand 2. The first one that we're going to talk about here, which will allow us to construct a microbial population growth curve, is something called serial dilution and spread plating. This is an example of what's called direct viable counting. So let's break that apart. By the way, in your textbook, there are several different ways of counting bacteria. I've chosen 4, and there's a variety, right? So direct, indirect, viable, total, right? So we get 1 of each. So direct means when I'm all done, I look and I can see bacteria directly. Does that make sense? There's a colony of bacteria. That's my ultimate goal, is to count a colony of bacteria. Right? So that means direct. Direct means I see bacteria directly. Viable means the only bacteria I see are alive and capable of growing. Right? Now that would be important, right? Because let's say like today, we're going to look at all of your motility media, and then we're going to put them in the morgue, and Emily is going to sterilize them for us. If someone looked at those media under the microscope, they would see loads of bacteria. But they're not a danger to anybody because they're dead. So sometimes it's very important for us not to know how many bacteria are there, but how many live bacteria are there, because it's live bacteria that can make you sick. Right? Dead bacteria are not going to make you sick. Live bacteria make you sick. Right? So being able to use a methodology that allows you to directly see only viable bacteria is very useful. As you can see from this, it takes quite a bit of work to make that happen. Right? So here's a culture. So you wonder how many bacteria are in that flask right now, right? Because normally you just look and go, oh, it's turbid. Yay, it's turbid. And then you look at it under a microscope and you go, yay, I can see them, right? But you don't know how many are in the flask. You just know that there are a lot of bacteria there. So how could you translate there's turbidity to a number? Well, the problem with this is, right, if there are enough bacteria to be turbid, there are bazillions of them. Now, you guys did your ubiquity plates, right? Some of you ended up with lots of bacteria on the ubiquity plate. And what happens when you have lots of bacteria on the plate? They get crowded, and then you can't count them, because there's so many of them that are lying all on top of one another, right? So we have to dilute the bacteria in order to see them, much like your streak plates, right? One thing that we're gonna be doing on Thursday is looking at your streak plates. You've been practicing streak plate for isolation, right? I gave you a mixed culture, you put it on a plate, and then you diluted it by spreading it out on the plate. You streaked it across the plate. And then ultimately you ended up with single colonies, right? What we're doing here is not diluting on the plate, but diluting in a series of broths and then putting them on a plate so we can count them. So this process starts with your flask. You take a little bit out of the flask, just a sample, right? It's like when you're making soup and you take a spoonful to see if it needs more salt, right? So you do a little sample, and that sample is representative, right? Like when you taste, right, you stir your soup, you taste it. If it doesn't have enough salt in it, the whole soup doesn't have enough salt in it because you just stirred it, right? So that spoonful is a representative sample of your soup. So you stir it up and you take a representative sample out. And now we have to take that sample and dilute it so that we can count it. Because if we take that sample out, right, so here's the sample, 100 microliters. Teeny, 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 right? 100 microliters is, we'll go over here so you guys don't feel left out. 100 microliters is 1/10 of a milliliter. It's a very small volume. And we want to take a small volume because we want to leave enough in the culture, right? So if you were going to sample your soup by drinking half the soup, would that be super useful? Because now you only have half the soup, right? So you take a small sample, right? Like a spoonful. Take a small sample so that the majority of the culture is still growing, right? So you take a small sample and you dilute it in water. In this case, you take 100 microliters and you put it into 9.9 mils of water. So you have microliters and mils, right? We go, so we convert the microliters to mils. So we have 0.1 mil and 9.9 mils, right? That's the ratio. There's this much culture to this much water, which means there's 0.1 to 10 mils total. Does that make sense? Because 9.9 plus 0.1 is 10. So I have 0.1 bacteria to 10 mils of water. We don't like having points there, right? So what can we do to get rid of the point? It's just math, like 4th grade math, right? If I want to get rid of that point, I can multiply that by 10 and that by 10, and I get 1 over 100. You with me? That means that in this test tube, I have a 1 to 100 dilution of my bacteria. Because I need to know what that representative sample is. I need a number for it, right? Not just a loopful. I need to actually measure out 100 microliters. So I have diluted the culture 1 to 100. Now, we talked at the very beginning of class about the fact that we don't like to use fractions. We like to use scientific notation. And scientific notation of 1 over 100 is 10 to the minus 2. I should make that look more like a minus. Those mean exactly the same thing. So when you see this test tube right here is a 1 to 100 or a 10 to the minus 2, right? Do you understand where they got that number? 100 microliters into 10 mils total is a 1 to 100 dilution. And we need to know that because we want to count. And to count, you have to keep numbers, right? Can't spread it out on the plate with the loop because there's no numbers there. It's a loopful. We don't know how much a loop is because your loops are not calibrated. So we put in 100. Now, if I were to take this, even though I've already diluted it 1 to 100, if I were to put that on a plate, if I were to take this and put it on a plate, even if I only put a little bit, say another 100 microliters, and I just put 100 microliters on here and spread it all around and give it the whole plate to grow on, and I put it in the incubator, guess what would happen when I got it out of the incubator? The whole thing would be covered. It would look like the primary zone of your street plate. And you can't count that. So this is too numerous to count. Some of you experienced that with your ubiquity plates. When you have so many colonies so close together, you can't figure out where one begins and one ends, and you can't count them. It's So we say, it's more than 300. So this is too numerous to count, which means I have to dilute further. So I take from here, right? And here, there's always magic. You think biology is boring. There's always magic, right? The magic, just like the magic of your streak plate for isolation, was that you never went back into the culture, right? You put bacteria on the plate and then you sterilized your loop and then you spread it out, right? You never went back in. You never go back in again. To further dilute this, right, just like if I had my plate and I sterilize and drag them out, right? Spread them out. How do I do that here? I go from here. Not from that one, but from this one. And I, from here, take 1,000. And you're like, well, now you're throwing more math at me, right? The reason I can take 1,000 microliters here is because I don't need to worry about this growing. It's that one that I needed to worry about growing. So I could only take 100 from there. So here I take a whole 1,000. And 1,000 microliters equals, what do you think? If 100 equals 1/10 of a mil, how many does 1,000 equal? A mil. Right? So now I took 1,000 and I still want to have 10 total. So if I take a mil from here, how much water do I need to have 10 mils total? 9, right? Not 9.9 anymore, 9. Because 1 plus 9 is 10. So I now have a 1 to 10 dilution. It's a different dilution. This one over here, 0.1 to 10 total. This one here, 1 to 10 total. If 0.1 to 10 is 1 over 100, which is 10 to the minus 2, 1 to 10 is 10 to the minus 1. And then just like with your streak plate, we sterilize and spread around and sterilize and spread around. You do the same thing serially. You keep diluting, right? But your dilution factor doesn't change. It's 1 mil to 10 total, 1 mil to 10 total, 1 mil to 10 total, 1 mil to 10 total. So if we think about this like this, the streak plate, this, right, is like the undiluted, right, the first zone. And this test tube is like the second one. And this test tube is like the third streak. And that one is the fourth. And by the time you got to the fourth, did you find individual colonies? Hopefully. You are going to have a test where you have to do that, so you better hope you did it. Right? But that was the goal, right? To spread it out. So our goal was the same here, spread them out. We just, rather than spreading it on the plate, we spread it here because we want to be able to count. So we have to keep track of our dilution. And the way that we do that, right, we have to keep track of these numbers. So notice this one says 10 to the minus 2. That one says 10 to the minus 3. And you're like, but it was a 1 to 10. So why is it a 10 to the minus 3? It's the addition of both dilutions. Because you already had 10 to the minus 2 and you further diluted it, it's minus 2 plus minus 1, and minus 2 and minus 1 equals minus 3. And minus 3 plus an additional minus 1 is minus 4. And minus 4 plus an additional 10 to the minus 1 dilution is minus 5. And minus 5 plus an additional 10 to the minus 1 dilution, right? Because we do this, and then we go from here, into here, into here, and we're accumulating the dilutions. The dilution is getting bigger and bigger and bigger, just like it is over here. Because just like here, where we never went back into the culture, we're never going back into the culture. Yeah?
For the streaking, are you streaking after every dilution?
No. We're not streaking. I was just trying to show that what we're doing in the test tubes is similar to what we did on the plate.
Okay.
Does that make sense?
So
we're not streaking. All we've done is diluted it. So here's the thing. Serial dilution spread plating takes a long time. Because we just did all this work and we still don't have a number. We still have to put them on plates. So you actually bring up one of the reasons why we don't like doing this, and you get to experience this in a week. because you have to do all of this, and then you have to plate all of them. Because only when we plate them can we see, right? Because we want to know, are they growing, right? Are they there? Are they viable? Will they grow? In order to see if they grow, just like when you did your streak plate and I said, don't throw it away. You don't know how bad it is until you let it grow. You don't know what's going on until you let them grow. Yes?
Yeah, so like, that's what I was asking, if like, when it comes to plating them, are we plating each dilution or just the last dilution?
Yes, no,
Each
so what we will do is we won't do him because we know, just like the primary zone, there's gonna be too much. But we will plate this tube onto this plate, this tube onto this plate, this tube onto this plate, and this tube onto this plate. But not with a loop, because we want to be able to count and we have to have numbers. We're actually going to put a volume in there. We're going to measure out and transfer 100 microliters from this tube into this plate. So we know exactly how much, what fraction of that total we've put on a plate. Now, the thing about plating 100 microliters is it actually further dilutes the culture. And I'm not doing that to you because I dislike you and I like to make life confusing. The reason I'm doing that, right? Let's erase it. The reason that I'm doing that to you is really for your benefit. If you were to take, so if we have this 10 to the minus 3, and we put 100 microliters, or 1,000 microliters, right, or 1 mil onto this plate would still be 10 to the minus 3. But you would have to sit in the lab for like 3 hours waiting for that mil of fluid to soak into the plate. Does that sound like fun? At the very end of the day, you have to sit for 3 hours, because I'm not going to do it for you. So you're like, well, that sucks. I don't want to wait for 3 hours. What can I do to make it so it doesn't take 3 hours? You can plate 100 microliters, and it will absorb almost instantly. That's a better option, right? But when we plate 100, let's see this in a different color. When we plate 100 microliters, we've increased the dilution again because we're using 100 microliters. So now it's a 10 to the minus 4. Confusing math, but it means that you get a shorter lab day. And your lab day is already long, because guess what? This whole thing, taking out an aliquot, diluting it in tubes, plating it, that's one series. You do it 4 times. So all of this, and then you wait a half hour, and you do all of this again, and you wait a half hour, and you do all of this again, and you wait a half hour, and you do all of it again. Because we need to watch it grow over time. This is one number. This is how big the population was at 2 o'clock. How big is the population at 2:30? How big is the population at 3? How big is the population at 3:30? So all of this is done, right? So you take that 100 microliters out, spread it around, and then you're still not done, because what do you have to do in order to let them grow? You gotta incubate them. And then you gotta come, so you're gonna, it's gonna be terrible. You're gonna have to go home and not know what happened to your bacteria over the weekend until you come back on Tuesday. And then on Tuesday you come in and you go, oh, here's my plates, yay! And you get your plates out, and you go, you're like the 3 bears with Goldilocks, right? So Goldilocks rolls in and she's hungry, right? So she sits down at the table and she's like, this one's too hot. This one's too cold. This bed's too soft. This bed's too hard. This one's, it's kind of like that, right? So you pull these plates out, and the first one you go, I can't even count. It's too much. It's too numerous to count. And then you look over and you go, that's only 2. It's too few to count. So then you end up with this situation where you're like, well, that's garbage, and that's garbage, and statistically that's garbage, and this one is the only one that's just right. So you did all of those tubes, all of those plates, and everything but that is just stuff you did. This one is going to tell you how many bacteria are there, right? So you count these, and you can take it from me. I've actually counted. That's 159 colonies on that plate. Does that mean there's 159 bacteria in that test tube at that time? No, right, because remember, this is a representative sample, it's a tiny sample. So what we need to do is say, well, 159 that I diluted, so I have to undo my dilution. And here's where scientific notation is beautiful. This plate, right, let's see, we'll do this, there we go, we got green, right? So this was 10 to the minus 4, and we put 100 microliters here, which made this 10 to the minus 5. So I had 159 colonies when I diluted it, 10 to the minus 5. So how many are there? All I have to do is undo that minus sign. And in that culture, at that time, I had 159 times 10 to the 5 bacteria per mil. So, not right, and you go, okay, 159, 1, 2, 3, 4, 5, right? So 15,900,000 bacteria, not total, per mil. Lots and lots of bacteria, but taking metered volumes at every step allows us not to say, oh, they're diluted, like they would be on your street plate, but they were diluted this much, so this is how much is actually there, and it gives us a number that we can count. Now, in order to let anybody who ever reads our data know that this is a viable count, we're going to do something very special. We are going to report this as 159 times 10 to the 5 CFUs per mil. We're not going to say bacteria or cells, because we want everyone to know it's not just cells, it's viable cells. It's cells that are capable of growing into a colony, so we call them colony forming units. Whenever you see a count of bacteria and it says CFUs per mil, you know that somebody did a viable count. Not total bacteria, but viable bacteria, right? So bacteria capable of making you sick or doing whatever it is that they're doing. Not total, not dead and alive, but only live bacteria that are capable of growing. So this method, serial dilution and spread plating, is great because it tells us viable cells. You can probably already imagine, and you will get to experience firsthand, the drawbacks of this method, right? It takes a long time, it takes a bunch of media, right? Those are drawbacks. Taking a long time, right? I mean, we're not in it just to injure ourselves, right? Like when something takes a long time, that's considered a drawback, right? So it takes a long time, it takes 5 tubes, 4 plates, and all of this stuff, right? So it's media hungry. But what's the benefit? It tells us the viable cells. So your book will have other methods that are direct viable. There are lots of them out there, but the only one that we're paying attention to is this method here, because this is the one that we're going to be doing in class. You are going to be performing serial dilutions and spread plating, and we do it 4 times so that each of you gets to do each part. So everybody gets to do dilutions, everybody gets to do plating, everybody gets to count, so everybody gets a chance to do all the parts, right? So this method, right, it's a pro because it's viable. It's a con because it takes a long time and has a lot of stuff that you need, right? And you only have to do half of it. You have to inoculate the media. Somebody else has to clean it all, right? It's not fun. So when you think about these 4 methods that we're going to be looking at, think about what's good about them, what's bad about them, right? So what's good about this is we actually get a viable number. What's bad is how long and how much it takes to get us to that number. So that's direct because we're looking at colonies. And it is viable because they are alive. Now, incidentally, this right here, 159 times 10 to the 5, if we did count this one, you're like, well, why is that 159 and that's 17? Well, think about the math, right? This is 159 times 10 to the 5, so 159, 17, but what was that plate diluted? It was a -5, which increased to -6, so it's 10 to the 6. And if we correct, right, we make these the same, if we make them both 10 to the -6, we can move this decimal over and add it here, and I would get 15.9 times 10 to the 6th, and 17 times 10 to the 6th, which are pretty close. Which they should be, because they are both representing that same flask at the same point in time. Just like this one would be 2 times 10 to the 7, which is 20 times 10 to the 6. So so you did a good job diluting. So I can check and see if you did a good job diluting, because if you did, then they will all be pretty much the same number. If you did a bad job, then they'll be crazy and they won't make any sense. So that is a direct viable method. So how about a direct total method? Direct in that we're looking directly at bacteria. Total in that now we're looking at both dead and living cells. Viable and non-viable cells. Cells capable of reproducing and cells that are not capable of reproducing. So to do this, you can do a microscopic count. I want you to think about your wet mount, because that's the closest thing you've done to this so far. You buy yourself a fancy microscope slide. They're like $400. You put some media there, and it will spread the media out to a nice thin film that you can quantify the area in. And then you start counting bacteria. Now, half of you would have an easier job of this because you have non-motile bacteria. But still, we have Brownian movement and current flow, which makes any bacteria difficult to count, right, to watch. Because they move, right? If I'm supposed to count this box right here, and there's a flow of media through the slide, it's hard to count them. Now, add to that motile organisms. And they're spinning, and they're moving, and they're swimming, and how do you count them, right? So you might look at this and go, oh, that would be much better, that's much better. Like, all I need is a microscope slide, I put it under the microscope, I count it. So the pro of this one, the advantage of this one, is that it's fast, and all you need is a microscope slide. The disadvantage would be that if from a liquid culture, or even from a solid culture, if you ever put it on a microscope slide, you've got to add water, right? Which means you have a liquid culture which is moving, and it's more difficult to accurately count. The other drawback is that it tells you total cells. You can't tell whether they're alive or dead. So notice where the serial dilution and spread plate gave us CFUs per mil, this one is reported as cells per mil. So when you see cells, right, that actually, there's a distinction there that you probably never thought about. The distinction is, we don't know whether they're alive or dead. So we say cells per mil. We know these are alive, so we say CFUs per mil. So it's not just me being nitpicky about words, right? It's so that when you look at data, you can tell from those data, oh, that's a total count. Those are alive and dead. I may not need to be worried about that because some of them are dead. But this is a CFU. Those are all alive, able to grow. That could be a big problem, right? So the direct total method that we're looking at is microscope counts. But see how much quicker I could get through that and explain it to you? Because it's a far less complicated procedure. You just put them under the microscope and count them. So it's easier, it's faster. The drawback is, it's like trying to count small children. Do you ever try to count, like go on a field trip with a bunch of little kids, right? Like my job when I was in college was like day camps and after school programs. We would take kids to Magic Mountain, and then you had 20 kids, and you're like 1, 2, 3, 4, 5, right? And you'd be like, stand still, hold hands, right? Or you do that cool thing where you're like, you're 1, you're 2, you're 3, right? You make them count themselves, but you can't do that with bacteria. So when they move, you lost them. Now you're like, did I already count you? Or are you like 20? Are you 25? I have no idea, right? So the drawback is it's hard to get an accurate count. The advantage is that you can do it rather quickly and get just a general number. All right, so indirect viable, direct total, indirect viable. So viable means what? Live. Indirect means I see something and I assume that something increases just like cell number increases, right? It would be like if you all walked into the room and you were sweating and red-faced. I could assume from that it's hot outside. I don't know what the temperature is, I'm looking at something about you and I'm deciding something about something else. That's an indirect way of telling whether it's hot outside. If you're sweating and you're red in the face and you're sweating, then I'm going to assume it's hot outside. That's an indirect way. I could also, look on my phone, see what the temperature is. I could go outside myself, and that would be direct, right? If I looked at a thermometer, that would be a direct way of telling what temperature is. The problem with any indirect measurement is, if I assume that you walking in the door red-faced and sweating is because it's hot outside, there are other things that could make you red-faced and sweating. You could be going through menopause, which will make you red-faced and sweating. You could have gone outside and done 100 jumping jacks, which will make you red-faced and sweating. So there are other things that can account for what I'm seeing. So any indirect method is indirect because I'm looking at something and assuming that thing is the same as growth. So in this example, we inoculate media and we look for turbidity. And we assume that turbidity means bacteria grew. So viable, because we assume they grew. Indirect, because I'm not looking at bacteria. I don't see colonies, I don't see cells. I see turbidity, and I assume that that turbidity has increased because the cell number has increased. Right? But, there could be other things that would account for turbidity. I could have gone and sprinkled dirt inside all of your tubes, and you'd be like, oh, it's cloudy. There must be bacteria there. But they're not. It's something else. So any indirect method says this thing is increasing because the cell number is increasing. So the indirect viable method that we are looking now is called most probable number. And the most probable number is a very common test that's used to get a general idea of bacterial contamination of water and food. And what happens is you take a water sample, for instance, and we're going to get to do this in a couple of weeks in lab. You can even bring in a water sample if you would like to, right? Not your dog bowl, because everyone knows that's disgusting, right? But you're going to take water, and you're going to put, let's say, 10 mils in this set of tubes, 1 mil, which is a tenth of 10 mils, into this set of tubes, and then 0.1 mil, which is a tenth of that, into that set. So you're going to do this serial dilution thing again. And then you're going to take those 15 tubes, but I'll only make you do 3 tubes because I'm nicer. So 9 instead of 15. And you put them in the incubator, and you come back in 2 days, and you look for turbidity. So in the first set, all 5 of them turned turbid, right? So they're yellow now to show us turbid. In the second set, 3 of them did, and in the third set, 1 did. So you would read that as a 5-3-1. But that still doesn't tell you how many bacteria are there, right? What does 5-3-1 mean? Now, it'd be great if it meant 531, but it doesn't. So in order to figure out what 5-3-1 means, you have to access what's called a most probable number table, which you don't have to memorize. You can Google it. And when you go on that most probable number table, you take that 5-3-1, and you find 5-3-1. And now, your most probable number of bacteria, right, viable cells, so colony forming units per mil is 110. You're like, cool, that's a great number, 110. But most probable is like when I tell my kids, you could probably have ice cream after dinner tonight. It's not a guarantee, right? If it were a guarantee, I would say you can have ice cream after dinner tonight, right? They hate it when I say maybe and probably because they're like, I already know, right? So probable number doesn't mean the actual number. So let's say that we were like, okay, listen, you're going to tell me exactly how many bacteria are there. I don't want this maybe 110. I want you to be 95% sure that you're telling me the right number. And you're like, okay, all right, fine. If I gotta be 95% sure, the actual number of bacteria is somewhere between 34 and 250. Not a super helpful number, right? So the most probable number is great because it's pretty easy. Just put it there, bring it out, get a table, and you're like, I got a number. But you can't be really sure of that number. So there's a lot of wiggle room in that number, right? So the pro is it's pretty easy, right? It does take a long time. The con is you don't get a number you can be really sure of. And this is why we always pay attention to statistics, right? Don't look at the number. When they're like, oh, this drug, shrunk tumors in 57% of the patients. You're like, but be 95% sure of that number. And they're like, yeah, it's somewhere between like 0 and 100% of the patients. That's not good, right? That means it probably didn't do anything at all. So you don't just look at the number, right? The number will wow you, but you gotta look at the statistics. And then you see that the number's really not all that impressive. So why do we do an MPN? Because it's called a screening test, right? We screen it, and if there's any bacteria in your food at all, guess what we don't do? We don't eat that food, and we test it further. So we call this a presumptive test when we're talking about food and water safety. We have a presumptive number of bacteria, and then we test it to make sure. So like after we have a big rain, which hardly ever happens, which makes it even worse, after we have a big rain, they actually go out to the sewage outflows and they collect water samples and they do MPN analysis on them. And if they find any bacteria, guess what happens to the beach? It gets shut down until they confirm whether or not that number is high enough to be dangerous, right? So most probable number, viable, because turbidity we're saying is growth, but indirect because it's turbidity you're assuming is growth, not actually seeing a colony of bacteria on the slide. We will be doing that, but it will not be what we do with the serial dilution and spread plating. What we will be doing with the serial dilution and spread plating is everybody's favorite way of counting bacteria, and that is using a machine called a spectrophotometer. So this is a machine that measures light. So this is a machine that can actually quantify turbidity. You look at turbidity and you go, that looks turbid. That looks not as turbid. That's like going outside and saying, it's hotter today than it was yesterday. That's not, when you would ask somebody, is it hot outside? Yeah, it's pretty hot. What does that mean? Is it hot outside? Yeah, it's 87. Everybody knows what 87 means to them. Right? Like my dad lives in the desert. Nothing is hot to him. It's 120. He's like, it's so nice out. Right? If he says it's 120 out, I'm like, oh, that's hot. If he says it's nice out, I'm like, oh, well, then let's have dinner out on the back patio. And I'm like, what are you, insane? Like, it's 1,000,000 degrees out here, right? So this takes that turbidity and quantifies it for us. It gives us a number, right? Because metry, meter, means number. So it tells us how turbid something is. And this is the one that you're going to be doing. And it's everybody's favorite because all you have to do is take a sample and put it in a machine and it gives you a number and you write it down. Which is great, right? So we have these fancy machines, and the machine has a light source and a meter. And when there are no bacteria, all the light shines through. That's what you mean when you say that it's not turbid. You can see through it. But as the bacteria increase, that increases the turbidity, and now the light gets blocked, and so it says, oh, it's absorbing light now, and it gives you a number. So where the MPN is just turbid, this one gives you a number. It's not just hot out, it's 87 degrees out. And that allows you to have a piece of information that you can plot on a graph and that you can compare to somebody else, right? Just like if you want to know whether it's hotter today than it was yesterday, I mean, you could stand outside and then try to remember how hot it is and then stand outside again the next day. Or you could measure how hot it is today and measure how hot it is tomorrow. Now you have two numbers that you can compare, even though you got them independent of one another, right? So the spectrophotometer is indirect because, again, it's just turbidity. It's total because we didn't watch it grow. We're just looking at that, right? So it gives us a number, but that number is very useful because we can get it very easily. And it gives us a number so that we can compare it, right? So this is a very commonly used method of counting bacteria. And this is the one that you're going to use. So you're going to be doing serial dilution and spread plating of a culture every 30 minutes for a total of 2 hours. And you're going to do a spectrophotometric reading every 30 minutes for a total of 2 hours. Guess what's everybody's favorite part of that part? That one. You're like, oh, I'll do that all 4 times. And everyone else is like, no, you won't, because you're going to do the spread plating, and I'm going to do the easy one this time, right? So what that allows us to do, right, is compare different bacteria. So here are some bacterial cultures, right? So you go, oh yeah, that's not very turbid, that's a little more turbid, that's a little more turbid, that's a little more turbid, that's it, right. Or I can put them in a spectrophotometer and I can get numbers that allow me to graph those data. And in this instance, right, so this is some data, and it doesn't matter what, right? But these are data where we're looking at pH optimum of bacteria. So what pH do they like to grow best in? Now, assumably, over time, the pH they like to grow best in would be the one they grew fastest in. The time where their generation time is the slowest, or the fastest, right? So we let them grow for the same amount of time and put them in a spectrophotometer. So here's our controls, right? So this one, these are actually, this is my cute little bug that I grew when I was in graduate school, right? So they say you'll never use your stuff. You can. You can use it as an example in your class. That's the only thing I've ever used my data for. But here's my control, like, so here's him actually growing in filtered monolake water, and here's monolake water with nobody growing in it. Here's him at pH 11, pH 10, pH 8, pH 7, and pH 6. And which one do you think he likes best if they were all grown the same amount of time? What do you think? It's not a trick question. pH 10. He prefers pH 10. So that allowed me, right, and what do I want to do? I want to get out of graduate school. So what do you think I'm going to grow him at? pH 10. Because if I have to characterize him and figure out what he likes to eat and all those different things, I want him growing fast. So I did this as a preliminary experiment to figure out what pH he likes, and then from then on out, that is the pH I grew him at. Right? Which is interesting because the pH of Mono Lake is really like 9.4. So 10 was a little bit higher, but he actually liked it a little bit higher. So spectrophotometry is very commonly used because it's easy and it gives you numbers. And numbers you can look at and see how they change over time. All right, let's take another break. We're going to take a longer break this time because you want to use the restroom.